So, we are going to start our new topic, which is reproduction in bacteria. In bacteria. So, first thing, this word you will find few words. It's like first is reproduction, which simply means making copy. Which means if this is one thing, you can easily make two things from this one. This means here population increases. Okay, this means copy of anything. It could be anything literally. Then you will find another term which is recombination. Recombination simply means the population does not increase, even not decrease, but they don't have any change in the population. Whereas this recombination simply means if this is the DNA and here is the DNA, and for suppose here they have genome here, so they will try to change from here, like they will try to change some part from here to there. It's like DNA molecule exchangement. DNA molecule exchangement. Okay, and you will see that no new molecule will form here, no new molecule, because here you will only see the change. And here in this reproduction you will see, if I am taking one mother cell, so here you can see it's making copy, and here you will see two more cells. So this means in reproduction population increase, copy of things is made. So I'm sure it's clear to you. Then I need to erase this all. Okay, so now, but our today's topic is about growth and reproduction. So first thing, as I just told you, reproduction simply means making copy of itself. So we have two levels in this. Levels like what? This means first level which you see would be about protoplasm. Second which you will see would be number of cells. Okay, so it's like you will see in protoplasm they have the increase in size, and here you will see that it is increase in number, increase in quantity. Okay, I'm sure it's clear to you. Then this bacterial growth have four phases. So it's like these are on these basic, we have four other phases as well. So we have four phases. Then what these phases are, first you will see that would be lag phase. Second you will see would be log phase. Lag, log, two more as well. So a third one which you will see would be stationary phase. And the last one which you'll see I'm making here, that would be death or decline phase. Normal is called death phase. Okay, so I'm sure it's clear to you. So we are starting with the lag phase first. So in this lag phase, first thing that they increase in cell, like there is increase in size but no multiplication. This means size increase but no multiplication. No multiplication means no division. If I'm taking one cell, so it won't copy itself in these two cells, right? That's called division or multiplication, like that. Well, then here only size would be increased, and time, for sure time is required to make this type of adaptation. Time required. And they also synthesize new enzymes. And you will see that new enzymes will be formed, new environment will be there, and this phase you will see that there is high metabolic activities. then you will see that but still the cell won't be divided no matter if they have high metabolic activity, but cell will not divide, and enzymes will be formed as I just told you, and they will help in accumulating concentration, or they help in permitting their growth. Okay, so I'm sure it's clear to you. This is about lag phase. Then we have another phase which is log phase. This log phase is also called as exponential phase. And one more thing, maximum growth will only occur in this phase. What else? You will see that in this phase number of cell will increase with respect to time. Of course, time is required in all of these things, and number of cells will double with the cells double when double of time is provided them double time required. But for sure they can copy, they can make these type of things, but they require only time and average time required for population. You also see biomass, so average time which you see is would be known as double mass, that's called as generation time, and that's also called doubling time. This means more time is required, but they can for sure do a lot of things. Doubling time. This means cell multiply with the maximum rate. Multiply. And you also see that there would be linear relationship between logarithm of these cells. But for now, this is all. And yes, they also have more nutritions, and they also have toxic metabolic products. This means few bad things are also there. This means they produce toxic metabolic products, and you will see that they accumulate and inhibit the growth. So this is about log phase. Now we are log or exponential phase. Now we are going with another phase, which is third one, that is called a stationary phase. In this phase, you will see there would be a lot of nutrients and accumulation. Nutrients accumulation and formation of course, like nutrient would be formed and they will produce toxic product, which can even cause themselves as a death, or like they can even die from the toxic product which would be produced. And bacteria start the growth, and here bacterial growth gets complete. And you will see that there is multiplication rate, and they can also death can also be happen, and these all. So this is about this phase. Now we are going with the last phase, which is fourth one, which is called as death or decline phase. What happened in this phase? You will see that the progressive death will occur. Death will occur, and you will see that the rate of cell division will gradually decreased. Of course, if they are about to die and like they become on the position where there would be no replication, so the cell division rate would be decreased, and you will see that they stop multiplying. Of course, cell division will decrease, so this means they are not able to copy themselves anymore, and some bacteria also survive in spores or cyst during this phase, cyst formation during this phase. So I haven't reached the limit of this, but I'm sure it's clear to you. But still, one more picture you will see, which is in our book. Let me make another notes. So you'll also see that one picture is present. They simply tells that the here you will see time, and over this side you will see that the log 10, which is number of living bacteria, number of living bacteria. I could not write properly, but still, like, this is a graph present in our book. What will happen first, you will see that here till here you will see the first phase, which is log, not log but lag phase. I'm just telling you because here is a little mistake in our book. Here it's written log, but it would be lag phase, as it's our first phase. Lag phase will be continued, then here you will see log phase. Then when you move, stationary phase simply means a position where they don't do such things, but they become stationary. In physics even we have studied that they become on a particular, so this phase is called as a stationary phase. And in the last phase of decline, this means they fall down to the reach level, so here they are called as decline phase. So I'm sure it's clear to you. We are done with this video. Growth of bacteria is done, but still we have another part which would be binary fusion and these all types. I'll continue in next video. Till that, thank you for watching.